All right. Our last talk of the lightning session is from Alex McDonnell at University of Edinburgh, and we're going to see what yeast can do for these types of things. So, Alex.
Okay, thank you for the introduction. So my name is Alex. I'm a PhD student based in the...
A little closer.
It's okay. Based in the University of Edinburgh in the Kudla lab, and I'm going to talk about the project I've been working on for the past few years, which is looking at PAC6 DNA interactions using a combination of yeast-1 hybrids and, of course, deep mutational scanning. So our reason for wanting to study PAC6 in this way is it has quite a complicated genotype-phenotype relationship. So its function in the body is to act as a transcription factor that's essential for development, particularly in the development of the eyes, among other parts of the body. But when you get mutations in PAC6, particularly the more severe ones, so things like heterozygous gene deletions and nonsense mutations, you get a very distinct phenotype, which is demonstrated up here, which is this absence or underdevelopment of the iris, and this is known as aniridia. However, what's unusual is the vast majority of missense variants tend to cluster outside the disease spectrum of aniridia. As you can see summarized in these pie charts, you get this overwhelming enrichment of missense variants in the non-aniridia disease types. And so in order to investigate why this might be happening, we decided to do a deep mutational scan on PAC6, and this involved, very briefly, just taking a antibiotic resistant gene and cloning upstream of a PAC6 DNA binding target, and then integrating this into yeast, and then transforming in a PAC6 mutant library fused to GAL4, and then all of these yeasts were grown in a competitive growth assay, with the idea being that the level of disruption of DNA binding dictates the level of resistance and therefore the rate of growth. And of course, we measure this rate of growth using deep sequencing in order to generate the fitness scores. And so when we look at the functional consequences of introducing substitutions into PAC6, we see this recurrent pattern of positions which are pretty much intolerant to substitution, regardless of the nature of that substitution. And generally speaking, introducing mutation into PAC6 is fairly detrimental, which I guess isn't particularly surprising, especially when you consider how sequentially conserved PAC6 is in across organisms. However, what we do see is, particularly within the linker region between the two subdomains of this part of PAC6, we see a couple of, or 2 or 3 residues that have this massive increased inability to bind to DNA. And this is, I guess, particularly important when you consider PAC6's role in development. It has a very controlled level of expression and activity, and overexpressing PAC6 has been shown to cause ocular pathologies just like much as underexpressing it does. And so, in addition to this, we looked at taking these fitness scores that we generated and looking at their distribution when we group them by their mutant type. And you get a kind of result that you'd expect, where things like synonymous mutants and wild type tend to cluster fairly similarly, and the nonsense variants tend to be the least fit in the entire population. And between these two populations, you have most of the missense variants with multiple mutants tending to be more detrimental to function. And if we apply this same approach, but instead by grouping by PAC6 variants present in the ClinVar and Nomad databases, which are, as we've heard, kind of proxies for pathogenic and benign variants, we see a result that suggests that actually the vast majority of pathogenic variants in PAC6 are disrupting binding quite significantly, comparable to that of if you had no PAC6 expressed at all, such as seen in the nonsense variants. But what is also interesting to note is that this doesn't explain all of the variants in PAC6. So clearly there is more to the disease pathology than just PAC6 DNA binding. And with that, I'd like to thank you and welcome any questions.
Hey, how much variability is there in the PAC6 binding site as opposed to the PAC6?
Yeah, so one of the things I think that complicates the story is that PAC6's binding targets are quite variable. I mean, there is sort of a PAC6 consensus sequence, but when you look at things like CellX data, you see that like the actual sequences PAC6 bind seems to be very variable. And this is the follow-up question is this a kind of DNA specific fitness consequences that we're seeing? And so I've done, this was just for one DNA element, however I have done it with another DNA element. And generally speaking, the sort of the negative impacts we see on function seem to correlate fairly similarly, so it suggests that the same residues are being used in the binding.
I'm more interested if there are compensatory mutations in the binding sequence that would rescue, let's say, effects of the variants in the protein itself. Because I just assumed there's human variation in that sequence as well.
Yeah, short answer, I'm not sure.
Okay.
But there are examples of PAC6 binding targets that introducing just a single point mutation is enough to actually replicate the aniridia phenotype. So really quite severe.
Thank you.
Okay. And that brings us on time to the end of the lightning session. Future moderators take note. Tackling is an effective intimidation tactic. Thank you to our speakers. Maybe I can just have you all stand up one more time so that everybody can give you a round of applause. Stand up. I see you over there. Yes. Excellent. I detected also some other Canadians returning home from the United States like me. I've been your moderator for this, James Fraser, UCSF, and I'm out.